Okay, welcome to part 3 of this lecture where we consider the amino acids and how they are actually connected together to form structures that are called peptides. So we're going to talk about how amino acids are connected into peptides, which means we're going to talk about an important kind of covalent bond that does this job called a peptide bond. So amino acids bond to one another to form peptides. Peptides are chains of amino acids. The amino acids are the monomers of proteins, or the structures found within peptides. The monomer of a protein is an amino acid. The kind of chemical bonds that connect these monomers together are called peptide bonds. So peptide bonds are shown here, 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 and so on. And the monomers are amino acids. Okay? So peptide bonds form as a result of a condensation reaction. So peptides form by condensation reactions. They are broken by hydrolysis reactions. So if we think about what that means in very simple terms, for a condensation reaction, water goes out and the monomer goes in to the growing chain. In a hydrolysis reaction, water goes in and a monomer goes out. Let's look at a diagram of these reactions. So on the top we have condensation reaction, on the bottom we have hydrolysis reaction. We're focusing on peptides, but these terms can apply to many other types of reactions. Condensation reactions are very common forms of synthetic or building or anabolic reactions that occur in cells. Hydrolysis reactions are very common types of reactions that occur when you break something down, also called catabolism. So on the top we have a condensation reaction. The monomer, which is an amino acid, is being added to this particular region, and the result is the addition of an amino acid to this growing peptide chain and the release of a water molecule. So this is an energy requiring reaction. In contrast, if you were to break down that peptide, like if you had just eaten a burger or some soybeans for lunch and you were breaking them down so that you could get energy from them, you would have to put water in, and that would result in the breaking of this peptide bond, and you would get energy out as a result. So we'll be using the terms anabolism and catabolism a little bit more in later parts of the course. In this example, this condensation reaction shown is an anabolic reaction. This would be a catabolic reaction. So here's a little bit more chemically accurate depiction of peptide synthesis and the formation of a peptide bond. So we have an amino acid here and here. Okay. Note the structure that I just explained in the last portion of this lecture. Central carbon, hydrogen, amino group, carboxyl group, common to all amino acids. There is the R group. It's just a hydrogen in this case. So we have two hydrogens, that's lysine there. We have the same amino acid connected one to another. And these amino acids are going to form a peptide bond. The peptide bond is shown here. So the peptide bond always connects the carbon, shown in red, of the carboxyl group, shown over here, from one amino acid to the nitrogen, shown in blue, of the amino acid, or the amino group of the adjacent or next amino acid together. Peptide bonds always form at those locations, okay? Always. So as a result, when you have two amino acids connected together, or in fact a growing chain, you will always have them oriented the way that we see here, where there's an amino group on one end and a carboxyl group on the other. Notice that this was a condensation reaction, and water is always released as a result of condensation reactions. Also remember that this is an energy requiring or anabolic reaction. So here we have a much longer peptide. As a challenge question, see if you can go through this peptide and count the number of amino acids. Draw it out for yourself before you do that, and don't put the blocked or shaded coloring in. Just draw out all of the atoms and the bonds and see if from there you can determine where each amino acid is found. Once an amino acid is bonded into a peptide, it's referred to as an amino acid residue. So this structure shown here is also called the peptide backbone. It's very common terminology that you'll hear in science. So note that because of the nature of peptide bond formation, which always connects the carbon in the carboxyl group of one amino acid with the nitrogen in the amino group of the next amino acid, one end of the protein will have an amino group on it, that's over here, and the other end, I said protein a minute ago, but I meant peptide, the other end of the peptide will have a carboxyl group on it, over here. By convention, we refer to the group that has the amino poking out from it, the amino functional group poking out from it, over here, as the N terminus, and we refer to the end that has the carboxyl group as the C terminus. Also by convention, we usually draw these structures from left to right with the N terminus on the left and the C terminus on the right. Notice that when we draw them in this very flat, two-dimensional manner, all of the R groups have to point towards the bottom. And remember that in a cell, this peptide backbone, or this chain of amino acids, this peptide structure will actually be flexible and moving. And so it will be quite possible that it'll be somewhat twisted and that it'll be flopping around so that these structures can interact internally. Also notice that if you look at the drawing on the bottom in green, these three-letter codes like GLY and PRO, if you look at figure 3.2 of your textbook for reference, you'll see that they are an agreed-upon shorthand for the full names of the amino acids. Gly would be glycine, pro would be proline, and so on. Note that we also number amino acids, so we number them from the amino end to the carboxyl or C terminus end. So the first one, the amino acid number 1, will always be the one that contains the amino group that defines that end of the peptide as the N terminus, and so on. We always do that from left to right as shown here. The identity, sequence, and length of a peptide is referred to as protein primary structure. Okay? And again, this chain of peptide is referred to as a protein backbone. So we've just talked about protein primary structure, which is describing how a peptide forms and what a peptide is. So primary structure refers to the identity, sequence, and length of a peptide within a protein. Primary structure is dictated by the formation of peptide bonds, which are what connect amino acids one to another, and which amino acid is placed at which location in the peptide is dictated by the genetic code, which is the message written within the DNA of a structure called a gene in every single organism. In the next audio file, in the next lecture file, Part 4, we're going to talk about secondary, tertiary, and quaternary structure. Note that not all proteins actually have quaternary structure.